McParland. I'm a phytoliths specialist doing a PhD at the University of York, and I work for Historic England. I've been working on phytolith analysis, which is the study of plant silica microfossils from soil samples at Coppergate. These individual morphotypes are diagnostic of different plant species, and therefore I'm able to recreate the past environment and people's plant-based activities using these as a proxy. Phytoliths are plant silica microfossils. They essentially form when the plant absorbs monosilicic acid into groundwater, and during transpiration, when the water evaporates off, the silica hardens within the cells of the plant and forms a negative of that cell structure. So we'll end up with diverse forms like this one. I would first of all remove the clays from the sample by deflocculation. Essentially, you place the soil in a beaker and stir it so that all the clays float in the solution, and then you pour off the clays in the solution after all the silica and silt has sunk to the bottom. And following that, you would remove the carbonates from the sample by adding hydrochloric acid. You would then undertake organic removal by placing the samples in a muffle furnace, which burns off all the organics and leaves you with your silica microfossils. There will be other minerals within that though, so you then need to concentrate that silica into just silica, so getting rid of any other minerals. This involves the use of sodium polytungstate, which is a heavy liquid calibrated to 1.3 grams per centimetre cubed, which enables the phytolith to float and everything else to sink to the bottom. You then use a pipette to remove the phytoliths from the surface of the liquid and place it into a new tube. After you've completed this process, you dehydrate the phytoliths in alcohol, and then you're ready to put them on your slide and see what you've got. Well, this project has had two major aims. The first was to identify whether there were any phytoliths present in the samples at all. So in the UK, phytolith analysis is a relatively new technique. It's been used in the past a few times, but the lack of background research and understanding of which plants produce phytoliths and which plants don't has sort of held the technique back in this country, though it's progressed rapidly in many other areas of the world. So drawing on a phytolith reference collection which I have built myself, I've undertaken assessment of the samples to identify whether phytoliths were present, how many were present, and whether they were in a good state, whether they were degraded at all. All of this will help me to ascertain whether it's possible to do further work to identify those phytolith morphotypes to individual plant species. So it's not been possible during this project to do that because it takes many, many hours of comparison with the reference collection. And importantly, sometimes when I process samples from UK archaeological sites, I've not had any phytoliths. So it was an absolute joy to process the Coppergate samples and find that they're absolutely packed full of phytoliths. There are thousands of them. And for this project, I've looked at 3,000 phytolith morphotypes.